Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen, distinguished researchers, and esteemed participants of the 7th Borobudur International Symposium. My name is Eka Septiana, Magister Pendidikan, and it is an honor to present our study on behalf of my research team, Dr. Apoteker Kiki Herlianti, Master of Science, Apoteker Tris Harni Febriani, Master of Pharmaceutical Science, and Apoteker Sulistiowati, Sarjana Farmasi. We are from Sekolah Tinggi Ilmu Farmasi, Yayasan Farmasi Semarang. Today, I am pleased to share the result of our study entitled Evaluation of the effects of api-api fruit extract on hepatic and lipid profiles in rats induced with a high-fat diet and cigarette smoke. Let's begin. Cardiovascular disease and atherosclerosis remain leading causes of mortality worldwide. Both conditions are strongly associated with hyperlipidemia, chronic inflammation, and oxidative stress. Similarly, non-alcoholic fatty liver disease or NAFLD is increasing globally and is closely linked to a high-fat dietary patterns and exposure to toxins such as cigarette smoke. Because oxidative stress and inflammation are key mechanisms underlying these disorders, plant-derived compounds, especially phenolic antioxidants, are widely explored for their therapeutic potential. The api-api fruit, a mangrove species rich in phenolics, flavonoids, and other secondary metabolites, has drawn interest due to its anti-inflammatory and antioxidant properties. This biological potential forms the foundation of our research. The primary objective of this study was to evaluate the effects of api-api fruit extract on SGPD, SGOD, and total cholesterol levels in rats induced with a high-fat diet and cigarette smoke. These markers represent essential indicators of hepatic integrity and lipid metabolism, and their modulation provides insight into the therapeutic prospects of the extract. This slide summarizes the overall progression of our research. We began with the preparation of the ethanol extract, followed by phytochemical screening to identify major bioactive constituents. We then conducted an in vivo study which included the acclimatization of experimental animals, induction using a high-fat diet and cigarette smoke, intervention with various extract doses, biochemical measurements, serum collection, and finally statistical analysis. Each step was designed to ensure the reliability of the data and the validity of the outcomes. Our experiment consisted of 6 animal groups: a normal control, a positive control, a negative control, and 3 extract dose groups. Rats underwent 28 days of induction using a high-fat diet prepared from pork fat and quail egg yolk and cigarette smoke exposure. Following induction, 14 days of intervention with the api-api fruit extract were conducted. At the end of the intervention, serum samples were collected for analysis of SGOD, SGPD, and lipid profiles. This slide presents the extraction results. From 1.316 kilograms of wet Simplicia, we obtained 200 grams of dried Simplicia powder, resulting in a vinyl extract weight of 137.94 kilograms. This corresponds to a yield of 45.98%, indicating efficient extraction and suggesting a high content of alcohol-soluble secondary metabolites. On this slide, we present the phytochemical screening results of the api-api fruit extract. Several qualitative tests were conducted using specific reagents to identify the major secondary metabolites. First, the flavonoid test using magnesium powder, concentrated hydrochloric acid, and amyl alcohol produced a red, yellow, or orange coloration indicating a positive flavonoid content. The phenolic test with 5% ferric chloride resulted in a dark bluish color, confirming the presence of phenolic compounds. For tannins, the combination of sodium chloride and gelatin produced a wet precipitate which was also positive. Both Hager's reagents and Mayer's reagents yielded precipitates demonstrating a positive reaction for alkaloids. The saponin test performed by shaking the extract with hot distilled water resulted in a stable foam formation confirming saponins. Lastly, the Lieberman test produced a red to reddish purple color indicating a positive presence of 3 terpenoids. Overall, these results show that the api-api fruit extract contains a rich profile of bioactive compounds, including flavonoids, phenolics, tannins, alkaloids, saponins, and 3 terpenoids, which may contribute to its biological activity. This slide shows the phytochemical screening results for flavonoids, alkaloids, and steroid of triterpenoids. For flavonoids, the eluent used was N-butanol, glacial acetic acid, and distilled water in a 4:1:5 ratio. Ammonia vapor was used as the spray reagents. A yellow coloration after exposure indicates a positive result. For alkaloids, the eluent was ethyl acetate, methanol, and distilled water in a 100:13.5:10 ratio. Dragendorf's reagents was used as the spot detector, and brown or orange spots indicate a positive result. For steroids and triterpenoids, the eluent was chloroform, ethanol, and ethyl acetate in 9:3:5 ratio. Anisaldehyde sulfuric acid was used as the detector. Red or purple indicates 3 triterpenoids, while blue or green indicates steroids. All 3 tests gave positive results. Among the tested approaches, the high-fat diet plus cigarette smoke showed the highest increases in triglycerides and cholesterol. These induction parameters were therefore used for the full study. Notably, triglycerides increased by 23.47%, cholesterol by 22.97%, and body weight by 9.77%, further validating metabolic disruption. Here, we present the main findings of the intervention. The api-api fruit extract produced a marked reduction in total cholesterol levels across the dose groups. This highlights its potential as a natural lipid-lowering agent. However, the extract did not improve SGOD and SGBD levels. In fact, although increases were observed, they remained lower than the negative control group, suggesting partial mitigation but insufficient hepatoprotective activity. This outcome indicates that while lipid metabolism was responsive to the extract, liver cell injury caused by the induction model may have been too severe or irreversible within the study time frame. In conclusion, the api-api fruit extract demonstrated significant cholesterol-lowering activity in rats exposed to a high-fat diet and cigarette smoke. However, it did not restore SGOD and SGBD levels, suggesting that its hepatoprotective effects are limited under these conditions. These findings support the potential of api-api fruit extract as a candidate for further development in managing hyperlipidemia. Further research should explore longer treatment durations, optimized dosing, and purified fractions to evaluate its hepatoprotective potential more truly. Thank you for your attention and interest in our research. We hope that these findings contribute meaningfully to the advancement of natural product exploration in pharmacy and biomedical sciences. We welcome any questions, comments, or suggestions from the audience. Thank you so much. Wassalamualaikum warahmatullahi wabarakatuh.